Welcome to this explainer. Today, we're going to completely change the way you look at your own body. You know, when we hear the word cell, it's super easy to just picture this simple, kind of static, microscopic blob floating around under a lens. But the reality? Oh, it is so much more than that. The cell isn't just a basic building block. It's a bustling, hyper-advanced, totally self-sustaining micro-metropolis. We're talking about a place with its own power plants, its own superhighways, waste management systems, and a complex communication network that would honestly put any modern city to shame. We're going to unpack exactly how this hidden world operates, step by step, as we explore this hidden city together.
So to really wrap our heads around this metropolis, we have to start with a pretty fundamental question. How did scientists actually discover this bustling, alien world hidden right inside our own bodies? Well, it takes us all the way back to a ridiculously young, incredibly observant English scientist named Robert Hooke. At just 27 years old, Hooke was looking at a simple piece of cork under an early microscope. He was just trying to figure out why cork works so well as a bottle stopper. What he saw through that lens, though, were these tiny, empty pores that looked exactly like a honeycomb. He called them cells. Now, what he was actually seeing were really just the empty cell walls of dead plant tissue, but that single, curious observation blew the door wide open to a completely new frontier of biology.
Section 1. Discovering the Hidden World. From early microscopes to cell theory.
To figure out the fundamental rules of this hidden world, we actually look back to the rapid-fire formulation of cell theory in the mid-1800s. We're essentially starting from the outside looking in, tracing how scientists transformed Robert Hooke's initial discovery into biological law. First up, in 1838, Matthias Schleiden establishes that, despite having totally different tissue structures, all plants are fundamentally made of cells. Literally the next year, in 1839, Theodor Schwann realizes, hey, the exact same thing applies to animals. These two guys gave us the first two tenets. All organisms are composed of cells, and the cell is the basic structural unit of life. But it was Rudolf Virchow in 1855 who swooped in with a final, crucial piece of the puzzle. He realized that cells can only arise by division from a pre-existing cell. A mother cell divides into daughter cells, passing down its genetic information, its DNA, to the next generation.
Okay, let's dive into this. For the longest time, scientists were totally stuck using light microscopes, which, they max out at a magnification of about 1,000 times. But the invention of the electron microscope, that unlocked a staggering 100,000x magnification. I mean, this was a total game changer. Suddenly, scientists weren't just looking at the outer walls of the cell. They were zooming straight down into the city streets. This extreme magnification finally revealed the incredibly tiny organelles operating inside, like ribosomes and individual proteins. It basically proved just how incredibly complex this micro-metropolis really is.
Section 2. Life, Death, and Immortality. The dramatic stakes of cellular life.
And this brilliantly illustrates something really important. Because a cell isn't just a tiny machine, it's a living entity. It grows, it reproduces, and under normal circumstances, it faces an inevitable death. When cultured in a lab, normal cells have a strictly finite lifespan. Their DNA literally shortens after multiple replications, which leads to programmed cell death. But nature occasionally breaks its own rules. Take a look at HeLa cells. Isolated back in the 1950s from a cancer patient named Henrietta Lacks, these cells are famously immortal. Because they're cancerous, they have this specific telomerase activity that actually stops their DNA from shortening, meaning they can divide and reproduce indefinitely as long as the conditions are right. HeLa cells from that one original sample are quite literally still being grown in laboratories all over the globe today. It's wild.
But honestly, whether they're mortal or immortal, cellular reproduction is just an absolute marvel of engineering. When a cell divides, it has to duplicate its entire genetic code. This cellular activity is so remarkably precise that DNA duplication happens with an error rate of less than one mistake for every 10 million nucleotides. Let that sink in. 10 million. Those are the basic building blocks of DNA. Just think about how many millions of genetic letters are in your body right now, and the cell is copying them with near-perfect, flawless precision every single time it divides.
Section 3. Powering the microplant. Energy and compartmentalized metabolism.
So, to maintain that insane level of flawless precision, the cell obviously needs a massive amount of energy. If we zoom in even further to check out the daily second-by-second operations of this metropolis, you'll see animal cells basically act as miniaturized chemical plants. They pull their fuel mostly from glucose, storing and using it as ready-to-go energy in the form of a molecule called ATP. But here's the real challenge. Even a simple bacterial cell alone is capable of hundreds of different chemical transformations happening at the exact same time.
So, how does it not just dissolve into total chaos? The secret is compartmentalization. The cell physically divides itself up into different sections, or compartments, with super-specific functions, because different functions absolutely demand different structures. For instance, the enzymes working in the Golgi apparatus are completely different from the enzymes operating over in the mitochondria. Strict compartmentalization makes sure the right reactions happen in the right place at the exact right time without tripping over each other.
And on top of that, this microplant is just incredibly efficient with its resources, using these highly regulated metabolic pathways. Basically, a substrate enters the pathway, a very specific enzyme sparks a key chemical reaction, and boom, the final end product is created. But here's the genius part. Through a process called feedback inhibition, that newly created end product actually loops right back around, binds to a hidden off switch on the original enzyme, which we call an allosteric site, and literally changes its shape to shut it down. The cell essentially tells itself, okay, we have enough product now, stop the assembly line. It completely ensures the cell never wastes its precious ATP energy.
Section 4. Movement and communication. Dynamic mechanical changes and signal transduction.
All right, so we've seen how the cell powers itself and regulates its internal factories. But a city can't survive in total isolation, right? It has to physically move things around. The cell is an always-changing, open system. It is not some rigid box. Just imagine specialized motor proteins acting like microscopic ATP-powered delivery trucks. They literally walk along the cell's internal highways, hauling vesicles from the endoplasmic reticulum straight over to the Golgi apparatus. And these highways? They're part of a cytoskeleton built from microtubule tracks that are constantly assembling and disassembling, adapting to exactly what the cell needs in that specific moment. The ground is quite literally shifting and rebuilding itself while the cell operates. You know, what's really interesting about this slide is how it perfectly explains the way the cell talks to the outside world, and why this directly impacts modern medicine. Cells are covered in receptors that listen for external stimuli, which is a process known as signal transduction. But exactly where those receptors are located depends entirely on the signal itself. Hydrophilic signals are water-soluble, meaning they can't penetrate the cell's fatty membrane, so they have to dock at cell surface receptors on the outside. Hydrophobic signals, on the other hand, can easily slip right through that membrane and use intracellular receptors on the inside. Why should we care? Well, because the vast majority of our everyday medications are actually hydrophilic. That means they rely completely on those exterior cell surface receptors to do their job and heal our bodies.
Section 5. The Master of Self-Regulation. Cellular Robustness and Biological Fail-Safes.
Up to this point, we've built up this picture of the cell as a complex, compartmentalized communicating machine. But here's the thing. When machines break, they fail. A cell, though, it's living. It's an incredibly adaptable, self-correcting entity. To really appreciate this adaptability, consider a dramatic question posed by the scientist Hans Driesch. What happens if you take a sea urchin embryo right at the very beginning of its development and you artificially separate its first two cells? Basic logic tells us that if you split an early embryo in half, you should just get two broken halves of a sea urchin, right?
But the experimental result totally defied logic. Instead of forming half an organism, each separated cell immediately recognizes the change, completely adjusts its internal genetic program, and grows into a complete, living, albeit slightly smaller, sea urchin. This is huge. It brilliantly highlights that early embryo cells aren't locked into some rigid preset path. They possess the full ability to completely self-regulate and adapt to extreme, life-threatening external changes. So the crucial point here is this concept called robustness. It's essentially the quality of being strong and protected from dangerous fluctuations. Because the cell is constantly using that feedback regulation we talked about, it is incredibly robust. Whether it's stepping in to quickly fix a dangerous DNA mutation before it can replicate, or instantly hitting the brakes on the glycolysis process when it senses plenty of ATP in the system, these biological fail-safes are the absolute only reason complex life can exist at all. Without this intense robustness, our little Micro-Metropolis would completely collapse overnight.
Which leaves us with this thrilling final thought. We know cells generally have finite lifespans, but we also know that activating an enzyme like telomerase can make them immortal, just like those HeLa cells. We know they are absolute masters of self-regulation. So, if a single microscopic cell naturally holds the blueprints for immense self-correction and even immortality, what limits might we break next with modern gene therapies? If we're currently learning how to rewrite the very rules of this Micro-Metropolis to cure genetic diseases, what is the ultimate ceiling for human biology?
It's such a fascinating frontier, and it all starts with understanding that hidden city inside of you. Thank you so much for joining me on this explainer, and keep questioning the microscopic world.